Welcome to our Microbiology Essentials series of tutorial videos handling bacteria. Today we're going to perform minimum inhibitory concentration testing. MICs are very important and will tell us the susceptibility of an isolate to an antibiotic. The MIC is the lowest concentration of an antibiotic required to inhibit growth. There are a few methods available, and today we are going to be using the microbroth dilution method in a 96-well plate. Let's begin. First, we are going to prepare 2 tubes of sterile growth media. In our case, this will be cation adjusted Muller-Hinton broth, which is suitable for antibiotic susceptibility testing. 1 tube will be for the culture, and the other will be a media control. Select a single colony of your isolate. With a sterile loop, pick up a well-isolated single colony, taking care to not touch any nearby colonies. Transfer the colony into the liquid media. Now it's time to label the tubes and incubate at 37 degrees overnight. Make sure that they will be continuously shaken. That's all for the first day. Returning the next morning, collect your tubes from the incubator. There should be visible growth in the culture tube, and the media-only tube should be clear, as it was the day before. If it's not, then that means that some contamination is present, and we can't be confident that our culture is what it should be. If that's the case, we would have to start again. This is a typical bacterial growth curve. It can be broken down into 3 stages: the lag phase, exponential or logarithmic growth, and stationary phase. After an overnight incubation, our culture will have reached stationary phase. In this stage, there will be a mixture of dividing cells, dead cells, and cellular debris, all contributing to the cloudiness of the liquid. We need to get all our cells into early log phase so we can accurately estimate the number of cells we will use for our MIC and get a standardized result. Early log will be in this range, around 0.1 to 0.3, measured in a spectrophotometer at 600 nanometers. Let's make up another tube of sterile growth media and transfer a small amount of our overnight culture. This new culture will take a couple of hours to reach early log phase, so this will give us plenty of time to prepare antibiotics and set up the 96-well plate for the assay. Make sure the amount you transfer isn't enough to make the media go visibly cloudy. Something in the range of 10 microliters in 10 milliliters of broth should be fine. Let's put it in our shaking incubator at 37 degrees and move on to setting up everything else we need for today. If you don't have a rough idea of the expected level of sensitivity of your organism to your drug, it is generally a good idea to do 2 sets of MICs, 1 high and 1 low concentration. Here we are going to use a low concentration. As we have already predicted, our strain will be quite susceptible. It's important to adjust concentration calculations for the potency of the antibiotic you are using. Potency is expressed as a percentage of the activity of the antibiotic being tested against its pure form. In our case, we're using cefotaxime sodium salt. The molecular weight of cefotaxime is 455.5, and the molecular weight of cefotaxime sodium salt is 477.45. This means that our compound is 95.4% cefotaxime by weight. The supplier's stated purity for this batch is 96%. So, our potency value, calculated by multiplying 96% and 95.4%, is 91.58%. We will make up a stock solution of 4096 micrograms per ml. To achieve a concentration of 4096, we will need to prepare a solution of 447 2.5 micrograms per ml. We want roughly 3 to 5 ml of 447 2.5 micrograms per ml. So we will need roughly 20 milligrams of antibiotic powder. Don't try and weigh this exactly, we will adjust the amount of media we use to make up our solution. Any amount roughly close to the weight we want will be fine. Take care not to touch or move the vial. Okay, that will do. It's close enough. Now we can calculate how much liquid to use to dissolve the antibiotic. We want 447 2.5 micrograms per ml. and we have 13.78 micrograms, so we will need to dissolve in 3.08 millilitres. If your drug will dissolve in growth media, then it will be okay to use that, and it will make life a little easier later on. Mix well and make sure it's completely dissolved. Next, we need to sterile filter the drug. Using a blunt fill needle and sterile syringe, suck up the drug solution. Switch the needle for a 0.2 micron filter and transfer the solution to a fresh vial. Now we need to dilute our stock solution to the maximum concentration that we are going to use in our assay. In this case, it will be 16 micrograms per ml that we need. The final volume of each well in our 96 well plate assay will be 100 microlitres. 90 microlitres of this will be our drug solution with 10 microlitres of cell suspension added and diluting it as a consequence. Because of that, our drug concentration will need to be adjusted. To achieve a final concentration of 16 micrograms per ml in our maximum concentration wells, we will need a solution of 17.78 micrograms per ml. To get from 4096 to 17.78, we will do 2 dilutions. First, we will do a 1 in 10 to take us to 409.6. Mix well, and then dilute again to make 17.78. Let's make a large enough volume to easily cover our needs. We'll make 3 ml today. For that, we need to add 130.2 to 2869.8 microlitres of growth media. Okay, now let's set up the plate. Add 90 microlitres of broth to columns 2 to 11 and 100 microlitres to column 1. Now we can add 180 microlitres of our antibiotic solution to column 12. Our plate now looks like this, and the next thing we need to do is to serially dilute the antibiotic from column 12. We will take 90 microlitres from column 12 and mix it into the next column to the left. We will then repeat until we reach column 3, and then discard the final 90 microlitres. This creates our concentration gradient. Starting with column 3, each well to the right has a concentration double that of the one to the left. All wells apart from column 1 now hold 90 microlitres, and these are the drug concentrations in each column. All that we have left to do is to add our bacteria. It's time to measure the growth of our culture. We want an OD of between 0.1 and 0.3, measured in a spectrophotometer at a wavelength of 600 nanometers. Use growth media to blank the spectrophotometer and then measure the culture. Perfect. Now let's calculate how much to dilute this culture by to give us the correct inoculum for the assay. We have previously made a standard curve for this organism, and 0.2 OD will give us roughly 1 × 10 to the 8 colony-forming units per ml. The standard inoculum size for this assay is 5 × 10 to the 5 CFU per ml. We will be adding 10 microlitres of our culture to the 90 microlitres of media we prepared in our plate, so we will need to dilute our culture to 5 × 10 to the 6. Our culture is at OD 0.154. With reference to our standard curve, this will be around 7.7 × 10 to the 7 CFU per ml. So to achieve our target, we will need to dilute by a factor of 15.4. If we do that in a final volume of 10 ml, then that will be 649.4 microlitres of culture. It's simpler to measure out 10 ml and then replace 649.4 of the broth with your culture. Now we just need to add 10 microlitres of the cell suspension to every well in the plate, except for column 1, which will be our negative control column. The plate now looks like this. Every well holds 100 microliters. Column 1 has no cells. Column 2 has no drug. These are the negative and positive controls. Now we will incubate the plate overnight. Seal the plate with a strip of parafilm. All that's left to do today is to check that we have used the right number of cells. We are going to dilute our cell suspension, plate it on solid media, and grow that overnight. Tomorrow we will count the colonies and calculate the number of colony-forming units per mil. Using a fresh, sterile 96-well plate, we will add 180 microliters of PBS to 5 wells. Next, we will add 20 microliters of our cell suspension to the top well. That's a 1 in 10 dilution. Now we will take 20 microliters from that well and add it to the next one down, and so on until we get to well 5. We are now going to plate out 20 microliter droplets. If our cell suspension is the right density, we would expect to see around 100 colonies in each droplet at a 1,000-fold dilution, and 100 per droplet in a 10,000 dilution. We will plate out droplets from rows 2 to 5. Let the droplets dry, then flip the plate and incubate overnight. Collect the agar plate of droplets from yesterday. Each section of the plate should have a 10-fold difference in the number of colonies from its neighbours. Select a dilution with 30 or more colonies and count them all. Now we can calculate our starting inoculum. In order for the MIC to be valid, we need to have inoculated the plate with the correct amount of cells, 5 times 10 to the 5. An acceptable range is 3 to 7 times 10 to the 5 CFU per mil. We can calculate our starting inoculum density by multiplying the average number of colonies we counted by their dilution factor, then dividing that by the droplet size in microliters, and multiplying by 1,000 to give us CFU per mil. As we used 10 microliters of our inoculum in a final volume of 100 microliters, we need to divide this number by 10. If this falls within our acceptable range, then the MIC is valid. Let's get the results. Check the negative control column is clear and the positive control column is cloudy. Then look at the rest of the plate. There should be conformity between rows of replicates. Hold the plate up to the light and look at it from the underside. Any well with any cloudiness has grown. We are looking for the column at which the media is completely clear, with no visible growth, the same as the negative control. This will be the concentration which inhibits growth. This is our MIC. That's all for this video. See you on the next one.